Okay, so now in this video, we get to actually talk about how a muscle contracts. So remember, we have our two main filaments that we talk about. We have the thick filament, we have the thin filament. Remember that our thick filament was made up of a filamentous tail, and then it had this big globular head on it, right? I'm just going to draw one for the sake of simplicity right now. Okay, and the thin filament, right, was just that actin filament that had that sort of tropomyosin wrapped around it and then troponin attached to that. I'm going to draw it without tropomyosin and without troponin just so it doesn't get in the way. But for the sake of this, we're going to assume that calcium is present inside of the sarcoplasm and that that myosin binding site is open. Now, one of the things that we've sort of left out that we need to add in before we go any further is that under sort of a rested state, this myosin head has a molecule of ADP and inorganic phosphate attached to it. Okay, so you've got sort of ADP and inorganic phosphate attached to it. All right. Now, what's going to happen is that that myosin head is going to go up and it's going to attach to that thin filament. Okay, so it's going to go up and it's going to attach to that thin filament, like so. All right, now when it attaches, right, this is a chemical reaction that's taking place. So when that happens, it makes that inorganic phosphate that's attached to that unstable. And so it's going to release that inorganic phosphate. So what's going to happen is we have that inorganic phosphate gets released. Okay, so remember we've got ADP inorganic phosphate up there. That inorganic phosphate is going to get released. So what we have, right, we got our thin filament. Okay, and you have the attached thick filament, right? But during this, when we sort of release that inorganic phosphate, what's going to happen is it's going to cause that neck region of the myosin filament to sort of bend. So what's going to happen is that the myosin head is going to undergo a conformational change to where it looks like this. Okay, so that's going to be what we call the power stroke. So this step right here is a very, very important step. This step right here, very important step, because this step is going to be what we call the power stroke. Okay, now imagine that all of this stuff is sort of attached to other things. Right, so if we draw this in a line with one another, right, is that what's going to happen is when that myosin head goes from this conformation, right, and it switches and it goes to this conformation, like so, right, it's attached to that thin filament, that actin filament, right? So what happens is that that actin filament gets pulled in this direction, right? So it gets yanked. And so that is the power stroke, and that's actually the first step at where we see contraction of the muscle. Okay? Now, when it undergoes this conformation, remember that this myosin filament still has an ADP attached to it. Okay, still has an ADP attached to it. But when it undergoes this conformational change, it makes that ADP unstable. And so what's going to happen is that ADP gets released. Okay, so we have that ADP that is released. Okay, so then what we have is we have our thin filament. Okay. We have our thick filament, like so, with nothing attached to it. Okay, and so what's going to happen, do you know, is that in the next step, we're going to have a molecule of ATP come in. Okay, and when that ATP comes in, right, the next thing that happens is that we have our thin filament, like so, and when ATP comes in and it binds, is that the myosin head, oops, the myosin head is going to detach from that thin filament. So what's going to happen is the myosin head detaches from the thick filament, right? But notice it's still in this sort of, this conformation like this, right? So what happens is when that myosin head detaches, it makes this hinge region of that thick filament unstable, and so it undergoes another conformational change where it goes back to the original conformation, and ADP is hydrolyzed into ADP inorganic phosphate. And so notice, right, that we've sort of created a loop or a cycle. Now that cycle is really important because remember that the thick filament, right, is made up of multiple myosin heads. So I'm just going to sort of draw one thin filament, one long thin filament like so, right? And remember that we have a thick filament that's got multiple myosin heads on it, like this. We'll sort of draw them like that. Okay, now what's going to happen is that this one is going to go up, it's going to attach, and when that one goes up and attaches, right, it will go through that cycle that we talked about, and it's going to pull the actin filament in this direction. Now, before this one releases, this one will come up and attach. So as this one attaches, this one will release. So this actin filament, this thin filament, is sort of held in place, right? So then this one will pull on it this direction, and then this one can go up and bind, and it will pull in this direction again, right? And so you have this sort of walking along the thin filament. Now, what's going to happen is that when this one goes up and binds, and we pull this actin filament in this direction, it'll bind here on the first go round, and then it can come up and it can bind again in a secondary location. So if I were to draw the actin filament again after it's sort of moved, what's going to happen is now it can go up and bind in this direction, and it can continue to do this and sort of like walk along that actin filament so that the actin filament slides, or the thin filament slides along that thick filament. And this is what we call the sliding filament theory. The sliding filament theory. Okay? And that's how the actin filament slides along the myosin filament, or the thick filament. Okay, so remember the sarcomeric anatomy or morphology, right? Because in the next video we will look to see what happens to the sarcomere after this cross-bridge cycling, or as this cross-bridge cycling occurs.